For that lovely introduction, for inviting me. I've had a great time here, and for reading my papers. I really appreciate that. And what a great place. I've never been to Ann Arbor. I'm really impressed with the institution and with a university that has a mammal for a mascot. That's great. All right, so sound okay in the back? Good. All right, so I want to give you a little perspective on the problem that I'll be talking about today. So imagine that the next time you're out at a meal, instead of eating the food that's on front of you in the plate, your dinner selection instead comes from the bouquet on the table. What would you choose? The orchids have terpenes that will irritate your mucous membranes. The calla lilies have tannins in them that will disrupt your digestion. And the poppies have mind-bending alkaloids. So what would you choose? I think that's pretty easy to tell for this audience, but this is the problem that mammalian herbivores face every day. Most mammals incorporate some plant material into their diet and are considered herbivores, but plants are not completely defenseless. They produce a wide range of detoxification compounds to deter herbivory. Let me give you an example that I won't get a chance to talk about later in the talk from our own work. This is Neotoma stevensii. It occurs in the desert southwest and is a specialist on the juniper that you see in the background. This animal consumes a diet of only juniper, and in doing so, it ingests a dose of terpenes, which are the primary component in turpentine, that if we scaled up to human body size would be 5 times greater than the lethal dose in humans. And this animal does this every single day. So one of the goals of our research is to understand how mammals like this are engaged in this battle with plants and their secondary compounds and seem to be winning. And so what I'd like to do for you today is highlight some of the strategies that we've been trying to identify in my lab. And I have 3 stories to present today. So I'll talk to you about the physiological adaptations that mammalian herbivores have for dealing with toxins in their diet, about the collaborations that they've established with microbial partners in their gut, and finally, some of the manipulative strategies like Mark highlighted in the introduction that they use to get around workarounds for plant secondary compounds. And the first 2 stories compose the meat of the talk. They're the longest sections, a lot of data and figures, and the last section is short and has videos. So if you're tired at the end, you can just zone out and watch the videos. So one of the organisms that we tend to focus on consists of the woodrats, also known as the pack rats. They're in the genus Neotoma, and these animals are perfect for dietary studies for a number of reasons. There are over 20 different species. There are specialists within this single genus. There are also specialists within a species. They eat a wide range of toxic foods, and because of their caching behavior and the fact that they occur in deserts, paleoecologists have uncovered their dietary history for us by radiocarbon dating their middens and identifying the plant material that's in their middens. And I'll be referring to this work in a few more slides. So these are a remarkable group of animals to work with. Today I'll be talking about the 2 species on the top, and I should say these diagrams are a little misleading. When you get out in the field, they all look the same. It's very hard to tell sympatric species apart, but I'll be talking about the desert woodrat, and actually not the whitethroat, but the Bryant's woodrat. So how do herbivores deal with toxins? And to study this question, we've been approaching it from trying to understand how herbivores undergo these major diet shifts in feeding on one toxic plant like juniper to feeding on another toxic plant like creosote. And I'll give you more background on this in just a few more slides. So let me set the stage for you by giving you some natural history. So the plant juniper currently is, and was a staple food for woodrats. This is a map of the desert southwest, in case you're not used to looking at that area of the world. And the green represents the historical range of juniper. And the green outline represents the current range of the desert woodrat, Neotoma lepida. So over 20,000 years ago, juniper was the staple food for woodrats as determined by paleoecologists. But then at the end of the last glacial maximum, the desert southwest was built. The southwest dried out, and creosote bush moved up from Mexico and invaded the southern U.S. And that range is shown here. So the red hatch represents the range of creosote currently, and you can see how it overlaps the range of the desert woodrat. So there are still populations like those indicated by this triangle here that feed on juniper and have never in their history experienced creosote. And there are populations in the southwest that currently feed on creosote. And this happened twice independently. So there is another woodrat, Bryant's woodrat, whose range is shown here in blue. And you can see that there's a small portion of Bryant's woodrat's range which penetrates into the Mojave Desert. And juniper and creosote are very different from a plant secondary compound profile perspective. So juniper is rich in compounds known as terpenes. These are the volatile oils that give conifers their smell. Alpha-pinene is the primary component of many species of juniper, whereas creosote produces a phenolic resin comprised of over 300 aromatic compounds. It's very complex, and it coats the leaves, and up to 20% of the dry mass of the leaves is in this resin. So this is considered a heavily defended plant. Very few insects have been able to colonize creosote. It's so toxic. Now, when I started this work, we began by focusing on liver enzymes. The liver of mammals is outlined here in blue, and that's because pharmacologists refer to the liver as the first line of defense against incoming toxins. So as dietary toxins are consumed, they're shunted to the liver through the hepatic portal vein, and they undergo first pass detoxification. They can often circulate in the blood and return to the liver, being detoxified after that. And I was naive when I started this work over 2 decades ago, because what I found was there are hundreds of detoxification enzymes, as listed here, in a single mammal species. So hundreds of enzymes, often induced by foreign compounds. And trying to figure out which enzymes to work on was a challenge. So we started by taking a pharmacological perspective, and we used the techniques that pharmacologists were using to understand drug metabolism in lab rodents, and that consisted of northern blots for messenger RNA of detox compounds, enzyme assays, and then finally, a few years ago, there was a breakthrough where we could survey a wide range of detoxification enzymes by using what's now obsolete technology, microarrays. So we built a liver transcriptome of woodrats, and we built a custom microarray with Agilent's help, and it's still available should you ever want to use this piece of technology. And so microarrays allow you to compare gene expression between organisms under different conditions. So we took this approach to understand how these animals were dealing with diets of creosote. And the way that we did this was we compared gene expression of our 2 species of woodrats in populations with and without experience to creosote. And this with and without experience to creosote will come up several times in my talk. So we collected these animals, brought them into the lab. They do very well on rabbit chow, and creosote resin is fairly easy to extract from the leaves, so we can coat rabbit chow with creosote resin and adjust the dose however we like. So we did this study, and we compared the gene expression of these animals to each other. And basically what we found was that experienced woodrats express more detoxification enzymes in their liver compared to the naive woodrats. So I'll just show you the results from one of the species, Neotoma bryanti, and the y-axis represents the percent of detox genes on the chip that showed higher expression. And you can see that a greater proportion of the genes expressed in the experienced animals are detoxification genes compared to the naive animals. So this suggests that the experienced animals have the machinery to deal with this incoming dose of creosote resin. And when we compared the experienced animals across these two species, we saw functional convergence in that the two different species were expressing two different types of detoxification enzymes, but they both serve the same function. So Neotoma lepida showed an upregulation of aldehyde dehydrogenase, and Bryantii showed an upregulation of aldo-keto reductase. And these both metabolize aldehydes. And aldehydes are one of the primary components of creosote resin. So the experienced animals are able to induce the appropriate detoxification enzymes when faced with creosote. But the naive animals tend to express genes related to stress, such as oxidative stress and cell death. So this suggests that our creosote experienced animals have adaptations for dealing with creosote. I just wanted to point out that we have tried to drill down and do structure function of particular detoxification enzymes, and I won't have time to talk about this, but we have been doing the structure function of one particular group of detoxification enzymes, the cytochrome P450s in the subfamily CYP2B. And I coursed a real biochemist who has studied this enzyme for his entire career to express this enzyme in his lab, and it made the cover of Molecular Pharmacology. So there's a non-model system on the cover of Molecular Pharmacology. And this work that we started with this biochemist actually informed his work on CYP2B in humans, because when he began to look at this molecule, while it doesn't seem to be important for creosote feeding as we originally thought, it seems to be very important for terpene detoxification. We are continuing to push this study forward with a study on the physiological genomics of these woodrats. So as Mark mentioned, we're very grateful for the funding that we got from the NSF, and I'm sorry, I'm sure you all hate me now, but we just got this funding to do this physiological genomics study, and my collaborator doing the genomics part is Mike Shapiro, and we're currently collecting woodrats with and without experience to creosote. We're collecting animals from those two species, Neotoma bryantii and Neotoma lepida, that have experience to creosote, that hybridize at their boundaries. So we're trying to collect some hybrids, or at least breed hybrids in the lab. And we have built a draft genome, and we'll take advantage of that and improve upon that to try to really drill down and understand of those hundreds of detoxification enzymes that occur in these animals, which are the important ones, and how have these two species independently evolved to be able to deal with this nasty toxin. This is where we're collecting from in California. Very interesting place to work. I told Mark that we were stopped by the Riverside County Sheriff, and I thought they were upset about where we were trapping, and in fact they were looking for a body, not for our woodrat traps. Luckily we didn't find the body. So I'm going to take a quick detour at this point and talk to you about something that we found along the way, and that has to do with the interaction of temperature with the detoxification of plant toxins. So we know that temperature alters the behavior of homeotherms like mammals. So here are woodrats shown at 21 degrees C and 28 degrees C in the lab, and you can see that their nesting behavior is very different under these conditions. And pharmacologists have known for over 50 years that ambient temperature affects the ability of an endotherm to metabolize pharmaceutical compounds. And that's true for a variety of pharmaceutical compounds that are natural products, like the ones shown here. And basically, at very warm temperatures or very cold temperatures, mammals are impaired in their ability to detoxify. So we wondered if this was the case in nature. And we've coined this phenomenon temperature-dependent toxicity. And this is the work of a newly minted PhD in my lab, Patrice Kernath. And what Patrice did is she collected animals from creosote feeding areas, and she exposed them to cool and warm temperatures on both a short-term and a long-term basis, and I'll just show you one of those profiles. And then she used hexabarbital as a proxy for liver clearance time. So hexabarbital is a sleep agent, a hypnotic agent. It puts the animals to sleep when we inject it, so we can measure how fast it takes them to clear this compound just by measuring how long they sleep. It's a very non-invasive process. So you can see in the cool, the animals clear hexabarbital much faster than they do in the warm. So this suggests that even though the animals are at the same body temperature, like their body temperature is not changing, the ambient temperature is influencing their ability to detoxify. She did another study where she looked at the maximum amount of creosote resin that these animals could ingest by putting them on trials and gradually ramping up the amount of toxin in their diet. And you can see that the maximum dose of toxin ingested by the woodrats at cool temperatures was much greater, almost twice as great as that at warm temperatures. And I should say that these temperatures are both temperatures that animals experience in nature, and the warm temperatures are within their thermal neutral zone. That's supposed to be their temperature sweet spot. And the cool temperature is just below their thermal neutral zone. So then we wanted to see what would happen if we fed them the same dose of creosote resin. So we picked a dose that was less than the maximum tolerable dose at each temperature, and we fed animals the same dose of resin at different temperatures. And we measured their ability to remain in the trial by monitoring their body mass over time. So when our animals lose more than 10% of their body mass, they'll often die at body loss increments greater than that. So we pull them out of the trial at 10%. So the proportion left means the proportion that hadn't lost 10% of their body mass over time. And you can see at 21 degrees C, they all persist on this dose of creosote resin, but at warmer temperatures, both within the thermal neutral zone, all the animals, almost all the animals drop out by day 10. So warm temperatures really seem to challenge the animal's ability to detoxify plant secondary compounds. And we've been wondering, how common is this phenomenon? Is it common across most herbivorous animals? So I've been working with collaborators at the Australian National University who are funded by the National Research Council there. And we've been looking at these hideous Australian marsupials shown here, the brush-tailed possum and the ring-tailed possum, and doing similar studies with them by challenging them with hexabarbital. And these are the data for the brush-tailed possum. And you can see that the pattern is the same. In the cool temperatures, the animals clear hexabarbital faster. They sleep for a shorter period of time than at warmer temperatures. So we think this might be a very general phenomenon across mammals, and it might represent a trade-off between homeothermy, maintaining thermal balance, while simultaneously using the liver. So the liver is the largest visceral organ. Using the liver generates a lot of heat. At warm temperatures, animals might have to trade off use of their liver for not overheating. So they may not be able to eat a toxic dose of plant secondary compounds at warm temperatures. Oh, I just have a slide to show you that possums are also behaviorally affected by temperature. So here's our brush-tailed possum at 10 degrees, and here's our brush-tailed possum at 26 degrees. So you can see they have very different behaviors. We think that this suggests that climate change may make being an herbivore much more challenging, that animals will have to dial back the amount of food that they eat as the climate warms, and that there could be this tipping point. Mammals require less food at warm temperatures, so it may be that they won't be affected because they'll require less food as the climate warms, but there'll be some sort of a trade-off between the level of plant secondary compounds and the amount of food they eat. And it's just hard to predict how this is all going to play out. But we've seen range contractions of many herbivorous species over the last 100 years, and it could be because of this trade-off between detoxification and maintaining the right body temperature. All right, so now I'll switch gears and move into story number 2, and that has to do with partnering with microbes. Now, I told you just a few slides ago that the liver was the first line of defense for herbivores in dealing with toxic inputs, and that's not really true. So if you look at the esophagus here, it comes down into the stomach, and the gut is really the first place that toxins interact with a mammal. And we know from recent studies that the gut microbiome is hugely dense and diverse in mammals, that across the gut itself there are many different communities inhabiting these different regions of the gut, that there are trillions of bacterial cells in our own gut and the guts of other animals with millions of genes. So they far outstrip our own genetic power in genomic content. And this has been called a hidden organ and an additional genome. And these microbiomes have been shown to have many different functions. One that we know of for herbivores is fermentation. And animal scientists have known that the microbial community is essential to our agricultural species. But what we wanted to ask was, could our woodrats harness this vast microbial power in assisting them in dealing with toxic diets? So this work was pioneered and led by a grad student in my lab, Kevin Cole, who is now launching his own lab at the University of Pittsburgh. And Kevin started by studying this structure of the woodrat gut that I'm going to refer to as the woodrat foregut. So that's labeled number 1. And morphologists had noted that many herbivorous rodents had this sac before their gastric stomach, but they had puzzled over its function. And so Kevin took the lead on this and investigated whether it could house microbes. And he found that the pH of this region was conducive to microbial growth with a pH of 4.5. We also just recently did this big study on gene expression in the foregut to find out if it was somehow different than that of the foregut of lab rats. And after a lot of money and a lot of work, we found it's basically unremarkable and heavily keratinized. So that was sort of a disappointment, but we learned something about the woodrat foregut. So Kevin set out to look at the abundance and diversity of microbes in this foregut. And that's shown here on this slide on the y-axis. We have the number of cells per gram of contents, and I have the gut plotted across the x-axis here. And I want you to look first at area 4. So that's the cecum. That's the area that's known to house a lot of bacteria that do fermentation for herbivorous rodents. And you can see that it has a very high abundance, 10 billion microbial cells per gram of contents, and that the foregut is on par with that level of abundance. So this suggests that the foregut has a high density of live microbes. We also measured volatile fatty acid production, which is a signature of microbial metabolism, and found that the VFA production in the foregut exceeded that of the cecum. So it's a very microbially active area of the gut, but the residence time in that section is very short, and so it's probably not important for fermentation. Kevin did 16S sequencing to identify which bacteria were occurring there, and we found that it was a very diverse community represented by 7 different phyla, mostly Firmicutes, which was represented at a very high level. 80% of the microbes found there fell into that phylum. And this departs from what's seen in ruminants, where ruminants only have about 30% Firmicutes in their foregut. Both the Firmicutes and the actinobacteria have members of that phylum that are known to degrade hydrocarbons. So this suggested to us that the foregut could house microbial OTUs that could degrade plant secondary compounds. So to really drill down and look at the microbial function of the foregut, we focused on these creosote feeders in the Mojave Desert, and we used the system of adding resin to the diet. And I've already told you that resin contains hundreds of aromatic compounds. And we took a metagenomic approach where we did shotgun sequencing of the microbes in the foregut, and we compared metagenomes, so microbial community genes, in hosts with and without exposure to creosote diets. We used an Illumina HiSeq and 100 base pair paired reads and had an average of 52 million reads per gut. And then we classified these functions with MG-RAST. So we shotgun sequenced a lot of microbial genes and then classified their functions based on a database. And this next slide shows what we found. And on the top here, we have the microbial communities of host-fed control versus creosote diet. And you can see that those microbial communities clustered out by the diets fed to their hosts. And now I'm going to show you a heat map where genes that are more abundant are darker in color. And so you can see that there were a number of genes represented in this study, and for those that have the blue arrows and the bold, those are the ones that were significantly higher in the host-fed creosote diet versus control diet. But I'm just going to focus on these two functional categories, the stress response and metabolism of aromatic compounds. So we saw we saw more genes for the stress response and metabolism of aromatic compounds in communities from animals fed creosote resin compared to control diet. And this response is what's seen in humans given high levels of pharmaceuticals. So the microbial communities of us respond by producing more genes to degrade the incoming dose of pharmaceuticals. We wanted to drill down and figure out which microbial genes were important in this response. And so we did a lot of sequencing of those genes, and we found that there was a single gene, Aryl alcohol dehydrogenase. And this gene converts aromatic alcohols, which are also common in creosote resin, to aldehydes. And this gene is known to play a role in the degradation of the compound in a number of different organic compounds, like the one shown here. This is one of the primary compounds in creosote resin, so we think that aryl alcohol dehydrogenase may play a role in compounds like this one, NDGA. We also found by looking at the flanking regions of these genes that these were being produced by over 50 different microbial OTUs. So it suggests that many microbes were producing this gene, that this is really important to the microbial community in dealing with this incoming assault of dietary toxins. And we've been puzzling about whether there's coevolution between the host and the microbiota, whereby the microbes convert aromatic alcohols into aldehydes. And I showed you in the slides before that woodrats have detoxification enzymes that convert the aldehydes into something else. So there might be this coevolution between the host and its microflora. And we're trying to get more into that, but it's a difficult nut to crack. So do these microbes facilitate the ingestion of dietary toxins? And to answer this question, we disrupted the microflora of our woodrats by feeding them diets containing antibiotics. And we use neomycin. Neomycin is not absorbed by the host and primarily acts on the microbes in the gut. And this is another one of those graphs that shows the persistence of animals in the trial. And when we fed animals control and creosote chows with no antibiotics, 100% of the animals in these two treatments made it through all 14 days of the trial. When we started feeding animals antibiotics, even on control chow, so no toxins, some of the animals dropped out, but this difference was not statistically different from our no antibiotic controls. But when we layered on toxins and antibiotics, all of the animals dropped out after 14 days in the trial. And this was a very low dose of creosote resin, lower than the animals normally eat, but we removed their ability to deal with creosote resin by disrupting their gut community. And we also found that diversity of the community decreased, microbial diversity decreased. And in other work, we've suggested that microbial diversity is really a key in detoxifying toxins in creosote resin. Well, while we were doing all this, we were inspired by work that was going on in humans, looking at new treatments for C. diff infections in hospitals. And what clinicians were finding was that these fecal transplants were highly effective. If you took a stool sample from a healthy person and transplanted it into an infected patient, that they would recover from these nasty infections. Now, this is hard to convince humans to do, but our animals are coprophagic, so they eat. So food is feces to them, so, and we have them in the lab, so it's easy to feed them what we want. So we went out and we collected naive woodrats that had no previous exposure to creosote, so ecologically and evolutionary naive, and we collected our experienced animals from regions of the Mojave to serve as donors. We also collected naive animals to serve as donors to the naive recipients. And then we fed our recipients the feces of these donors, creating a naive animal with an experienced microbiota and a naive animal with a naive microbiota. And then we put them in a feeding trial. So again, this is one of those studies where we look at the percent remaining in the experiment over time. We gradually increase the level of creosote resin in the diet. And as a reminder, all of these animals are naive to creosote. They've never seen it before. And so over the course of the trial, the animals receiving the microbiota transplant persisted to a greater extent in the trial, whereas all the naive animals with the naive microbiota dropped out. So just by doing a fecal transplant, we were able to improve the ability of naive animals to feed on this toxic diet. So I was at a Gordon conference with Mark, and he can verify that my grad student really said this at this meeting. Kevin stood up and said, "This was the single best day of my graduate career when I went and collected the urine from the woodrats in this trial." And this will become apparent in the next few slides. So this is what the urine looks like from the animals with their native microbiota when they're fed creosote. So it's kind of cafe au lait colored. And when he collected the urine from the animals that are naive but have the experienced microbiota, the urine color is very different. So why does that matter? Well, this indicates that the liver is seeing a different set of metabolites coming in for detoxification, because everything that comes out in the urine has been processed by the liver. So we like to think of this as metabolomics by ecologists that are not Mark Hunter, who has all those fancy machines in his lab, but we actually did real metabolomics, and we found that there was a clear distinction in the metabolites produced by the animals with these two different gut microbiotas. So same host genetic background, different gut microbiota. So we've been trying to tease apart this connection between the host genetic background and the microflora. And what we've done is we've done some liver gene expression in these naive hosts that have different microbial communities. And so what I'll show you in the next few slides is the genes that were induced on a creosote diet by these animals with different microfloras. So genes induced on a creosote diet by naive animals with their native transplant, there were 42 genes more highly expressed in the liver. And when we compare that to animals with the experienced transplant, we saw only 24 genes more highly expressed. So this suggests that the liver of the animals with their native flora is working harder, that more metabolites are coming into the liver in this case than when the animals have a microflora from an experienced animal. And the types of genes that were expressed were detoxification genes, so many detoxification genes, and some overlap in between the native transplant and the experimental transplant. So this suggests to us that the microflora is pre-detoxifying, if you will, the incoming plant secondary compounds so that less plant secondary compounds get to the liver. So the experienced microbiota attenuates the liver's response to creosote. All right. Well, in the last few minutes, I want to talk a little bit about how herbivores have developed these workarounds to plant secondary compounds. And as Mark mentioned, for my PhD, I got to work on this charismatic lagomorph, the North American pika. The pika is really unusual in that it's an herbivore. It does not hibernate, but it lives in areas where plants are not available in the winter. So it spends most of the summer collecting vast quantities of plant material that it stores to feed on during the winter. And a single pika will collect 2 large garbage bags, 50 kilos worth of vegetation, to feed on over the winter. And when I started working on this project, someone else had reported, and this is why I began working on it, that the diet that the animals are eating in the summer differs from what they're storing in their hay pile. So these are pie charts representing the summer diet and the winter diet of pikas. And in the summer, they're very generalistic in their foraging behavior. So they eat a bit of clover, some grass, some flowers, leaves of many species. But in the winter, at the study site that I was working, they primarily stored the leaves and flowers of a single species, Acomastylus rossii. And what I found was that these plants were very high in phenolics, and that the phenolics degrade over the course of the winter. And during the course of the winter, the phenolic levels decrease to levels found in the summer diet. So it becomes as nutritious from that perspective as the summer diet. But these phenolics help facilitate preservation. So this collection of toxic plants allows the animal to make fewer trips into the meadow, which is very dangerous and contains many predators, such as weasels. And it preserves for a longer period of time in the winter. So I've been really interested in how animals manipulate plant secondary compounds. And a few years ago, I had the opportunity to work with some Israeli scientists on a BSF grant where we eventually studied 3 species of spiny mice. So let me give you a little natural history on this project. So there are 2 species of spiny mice in Israel, and they can occur sympatrically, the common spiny mouse and the golden spiny mouse. And they feed on sweet mignonette, shown here. And this work was led by a very observant graduate student, Mickey Simuni Blank. And we also studied a 3rd species that occurs on Crete that actually was transported there thousands of years ago. So it's originally from North Africa and was transported there by seafarers several thousand years ago. So the animal on Crete has not seen sweet mignonette in its ecological history. It's seen it evolutionarily when it occurred in Africa, but it hasn't seen it in the last 3,000 years. And these 2 species regularly interact with sweet mignonette. So why is sweet mignonette so important? Well, let me tell you a little bit about their toxins. It has a very clever toxin system. The sweet mignonette makes glucosinolates, and the activating enzyme of the glucosinolates are separated. So in the seed, there is an enzyme called myrosinase, which activates the glucosinolates in the pulp. So it's only when the seed and the pulp come together that it creates this mustard oil bomb that makes the pulp then toxic. And what Mickey found was that these animals have interesting workarounds. So I might have to go back here to operate this. So the golden spiny mouse, that's the fruit of sweet mignonette. And you can see those little black things in there. Those are the seeds. The golden spiny mouse consumes the fruit whole and has to deal with all the incoming toxic glucosinolates. And this is where Mickey's observation skills came into play, because I am sure I would have missed this. But what Mickey observed when she fed the common was that they, come back. Now you see that thing dropping out of its mouth, that's a deliberate discard of the seed there. So you'll see this again where the seed comes out the side of its mouth. And so what this animal does is it completely avoids activating the pulp because it spits out the seeds. And yes, we quantified this, and you can see that the common tends to eat the seeds, the fruit whole. The common spits out the seeds, the golden eats the seeds whole. And she found that the seeds that were spit out by the common were just as viable as seeds that had been digested by birds. So spitting them out doesn't damage them, and it could be a mechanism for dispersal. So the plant seems to have manipulated the seed predator into a seed disperser. Then we went to Crete, and we found that they did something completely different. So would anyone like to guess at what the Crete animals were doing? We brought sweet Mignonette into Crete, and we fed them to these animals that hadn't seen it in 3,000 years. Exactly. I think that's the only alternative left, right? So here they are digging out the seeds and throwing away the pulp. So this was a super fun project to work on, and not what we expected. We expected all these animals to be seed predators when we began this work. So I hope you've enjoyed hearing the different ways that herbivores deal with these incredibly toxic diets, physiologically, microbially, and behaviorally. And I'd like to say that we've only begun to understand the strategies that herbivores use. Like, this is just the tip of the iceberg, and I feel like we have so much more to learn. And I've told you a little bit about how our work has influenced studies on human systems with our understanding of categorizing CYP2B in woodrats that's led to a pharmacologist studying that in humans. And I think there's much more to be learned by investigating natural systems. So, I have to thank the large team of people that have worked on these studies over the years. So I haven't talked about everybody's work here, but everything is based on the shoulders of someone else. So these are my lab members, past and present, and I've pointed out the people that led some of the projects. And I'd just like to shamelessly recruit for graduate studies at the University of Utah and in my lab at this point. I hope there are undergrads in the audience that are interested in graduate studies. We are a general biology department that does everything from biochemistry to ecosystem science, and there are many opportunities. And this is what Salt Lake City looks like. So it's surrounded by mountains. This is the campus here. So I'd like to take this time to acknowledge all my funding sources and collaborators on these many projects, and thank you for your time, and I'd be happy to answer any questions. How many non-toxic plants? So many of the habitats that they live in are deserts, and in deserts plants are highly defended because leaf tissue is so valuable. It's so hard to make given the water limitations and nutrient limitations in the desert. So these rats mostly encounter toxic plants, and most plants defend themselves somehow from herbivory. Does that answer your question?
I'm just wondering how much intelligence comes into play when animals deal with plants. So there is a choice, has to be downtime. But in this case, we don't have a choice.
If they had a choice, I think they would choose something non-toxic, although we've done other studies that I didn't talk about that showed that sometimes they choose to eat plants like juniper, and the detoxification process seems to help them thermoregulate. So they would only choose juniper when they were in the cooler temperatures, And they would choose to eat less juniper in the warmer temperatures. And we figured that that must be adaptive, because why else would they be eating a toxic plant?
So maybe a similar question would be, for some of the plants that have these toxic compounds, are they differently distributed among different plant tissues? I'm thinking if there's a plant that's trying to extract a seed disperser, a vertebrate seed disperser, would they have, say, seeds or fruits that have differently distributed toxins so that they're actually able to extract enable the plant to help them out with the dispersal strategy?
Yeah, so the question for those of you in the back that might not have heard it is, do plants play games with their toxins to manipulate seed dispersers? And that's exactly what sweet mignonette is doing. And like I said, we started this work with the mice as thinking they were seed predators, to compare them to birds, which ingest the fruit whole, and the seeds pass through their digestive tract and come out unchanged. So sweet mignonette is manipulating its seed dispersers when it can, and chilies are well known for doing this. So chilies are dispersed by birds, which don't have a receptor for capsaicin, so they're unaffected by the heat of the chilies, but mammals chew the chilies and break the seeds and are not good seed dispersers. So plants are surely playing that game.
So the anti-temperature story is better with the microbiome story. It seems to me that different ambient temperature might have gone to the body temperature. So it seems to me that it's not the temperature directly in their detoxic ability, but rather the temperature in their response to the microbiome or something else. So do you think we could show that those animals that in different temperature, their microbiome change a lot, and that is consistent with the microbiome of the naturally versus the experiment?
Okay, so I'll try to paraphrase that. So what role does ambient temperature play on the microbiome? And that's a really interesting idea. We haven't looked at that. So we don't know how the microbiome changes. But
Because still the animal's body temperature doesn't change at all. That's my observation.
that's right. The body temperature doesn't change. The blood flow patterns change. So when animals are at warmer temperatures, they tend to shunt blood to the periphery and away from the visceral organs, because the visceral organs generate heat. And so in order to maintain thermal homeostasis, they play this game of shunting blood to the periphery so they can dump heat away from their body. And that makes their visceral organs less able to do the work that they do at cooler temperatures. But there could be an effect on the microbiome. Leslie? So the question is are the woodrats manipulating their microbiome? And that's a really good question. We don't know if they are, but they could be doing that. And woodrats often collect the feces of other organisms. We don't know what that's about. It could be, to try to get vitamins out of those feces. It could just be their collecting habits, but it may be a way to introduce new members into their gut.
Relying on animals to disperse the seeds, why do they put any toxins into the fruit at all? It would be better if they let them eat away.
So the question is, why does sweet mignonette put any toxins into its fruit? And it's to direct the proper seed dispersers towards the fruit and keep the seed predators away. So I showed you an example of a seed predator that had overcome its defenses. And another seed predator that manipulates the fruit, but it then became a seed disperser. So sweet mignonette manipulated a seed predator into a seed disperser by packaging toxins into its fruit. And it likely deters other seed predators from consuming this fruit. So mammals are really built for thermoregulation in the cold. Like they're really good at dealing with cold, and they're not so good at dealing with warm. Mammals have an internal heater, but they don't have an internal air conditioner. So they don't necessarily need to feed on a toxic diet, but it seems to be energetically beneficial if they have access to a toxic diet. They tend not to have rabbit chow in nature, so the studies that I showed you are artificial that way in that they often have to feed on toxic compounds. Other questions?
I'll ask the last one. You were very careful to use the word naive and experienced as opposed to adaptive. Do you know anything about, in particular the first study where we're looking at the enzymes expressed by the organism itself as opposed to by the microbiome, whether there has been natural selection for different enzyme production, or whether you can turn a naive creosote-feeding organism into an experienced one over time?
So the question is about genetic adaptation to toxins. And so from our work on P452B, there's a hint of natural selection in amino acid substitutions at substrate recognition sites that seem to be very important, but we don't know what the function of that enzyme is yet. Like I said, we think it's for metabolizing terpenes. So there seems to be genetic adaptation in that case, and we're hoping this physiological genomics study will let us really ascertain that. We haven't seen adaptation over time, but it's curious that these animals have been able to adapt to creosote in the last 20,000 years. And when we expose the naive animals to creosote within a single generation, we can't get them to feed on the same levels of creosote as their conspecifics in the Mojave.
Let's thank Denise one more time for her time.
Thanks for your attention.
Thanks for that.